disinfection is a process specifically dedicated to pathogens inactivation. It can be considered as the last barrier towards pathogen spread into the environment, since it is positioned as last treatment unit inside wastewater treatment plants before the treated effluent is discharged into surface water. When wastewater reuse practices are foreseen, it is even more important to assure the safety of reclaimed wastewater from a microbiological point of view to minimize both the risk for workers associated to irrigation systems and the contamination of crops, especially in case of raw consumption produces. A key point debated from researchers is the role of disinfection on controlling antibiotic resistance spread. In fact, microorganisms are able to survive to the exposure to one or more antibiotics in concentrations over the inhibition or toxicity thresholds, developing several resistant mechanisms, potentially determining the ineffectiveness of current antibiotic therapies and the increase in the health hazard posed by many infections. For this reason, antibiotic resistance has been indicated as a worldwide sanitary issue by World Health Organization. Urban sewage is an important source of antibiotic-resistant bacteria, being wastewater treatment plants hotspots for antibiotic-resistant bacteria and antibiotic-resistant genes diffusion in the environment, and disinfection can represent a valuable control measure against this hazard. Disinfection options rely on both chemical and physical disinfectants. Chemical disinfectants cause the lysis of bacteria cells, while physical disinfectants act differently, one from each other, however, not damaging the cell integrity. Among chemical disinfectants, we can list chlorine-based compounds, chlorine dioxide and peracetic acid, while ozone, which is considered a powerful disinfectant in the drinking water treatment field, is applied as oxidant and not as disinfectant in the wastewater treatment field. Ozone is a strong oxidant, promoting also the generation of hydroxyl radicals, able to oxidize refractory organic compounds, even present at trace level, after the biological treatment. This means that ozonation is highly recommended when the quality of the effluent has to be improved before discharge because of anesthetic characteristics, such as color or foam, or when the presence of toxic or potentially toxic compounds for the aquatic environment and human health needs to be controlled, as for contaminants of emerging concern, being about this topic an open debate on progress. Other chemical disinfectants exist, but they cannot be considered as established processes, since further investigations about effectiveness and drawbacks are still required. Among physical disinfectants, we have ultraviolet radiation and pressure-driven membrane filtration, referring to microfiltration or ultrafiltration. About membrane filtration, it is important to stress that they cannot be considered as disinfection processes as such. They display a disinfection action retaining microorganisms due to the membrane cutoff, permitting the exploitation of these characteristics for disinfection when already adopted in biological treatments, such as membrane bioreactors. Let's move now to a short description of each disinfection option. Chlorine-based compounds have hydrochloric acid as disinfectant active principle. The compound mainly used in Italy is sodium hypochlorite. Special attention has to be paid to the commercial solution grade, since it rapidly degrades during time, especially in case of not adequate storage conditions, as high temperatures and direct exposure to sunlight. This decrease implies an inactivation reduction, but also an increase of chlorate concentrations. Calcium hypochlorite, a solid commercial reagent, is more stable during time, but it is rarely adopted due to the higher complexity of the dosage equipment. In presence of residual ammonia, chloramines are formed, displaying a slower inactivation kinetic with respect to free chlorine, meaning that disinfection performance is strongly affected by the upstream biological treatment performance. The main drawback of chlorine-based compounds is the formation of byproducts, meaning products of the reaction of disinfectant with other compounds in wastewater. Free chlorine leads to the formation of trihalometanes, while chloramines could form nitrosamines with relevant environmental implications. Chlorine dioxide is not a chlorine-based disinfectant, since its active principle is dissolved chlorine dioxide. This implies a totally different chemistry in water, as well as different byproducts, which are chlorite and chlorate. In addition, chlorine dioxide doesn't react with ammonia, and its decay is not significantly affected by organic compounds, contrary to chlorine-based disinfectants. Anyway, being an unstable gas, chlorine dioxide has to be generated on-site, requiring a higher complexity with respect to the dosage of a liquid solution, making it less diffuse and adopted by wastewater treatment plant managers. Peracetic acid is a good alternative to wide diffuse chlorine-based compounds for some interesting features. It displays a wide spectrum of disinfectant action. It is less affected by upstream treatment performance. As chlorine dioxide, it doesn't react with residual ammonia. Last but not least, no toxic byproducts are known, and its ecotoxicity is limited with respect to the ones displayed by the other chemical disinfectants. All these characteristics have made peracetic acid more and more adopted as disinfectant for wastewater, not only in Italy but also worldwide. Peracetic acid effectiveness and validity has been confirmed also by a recent handbook published by the Water Environment Federation in 2020, dedicated to the application of this disinfectant. About UV radiation, its disinfection action is associated to the radiation absorption at 254 nanometers by cell's DNA, inhibiting the bacterial replication ability. This effect is anyway temporary, since when exposed to sunlight, many bacteria are able to repair the damage, coming back active, through a phenomenon called photoreactivation. So far, no disinfection byproducts are known to be produced by UV radiation disinfection, not displaying any ecotoxicity effect on receiving surface water. However, differently from chemical disinfectants, UV radiation action is time-limited, not assuring a protection far from the discharge point. This is an advantage from an ecotoxicological point of view, but it could be a constraint when reclaimed effluents are directly reused for irrigation. Summarizing, disinfection has a fundamental role in controlling pathogens diffusion into the environment, and even more in case of agricultural reuse. Anyway, disinfection is a trade-off process, meaning that each disinfectant has pros and cons to be balanced. It is therefore of paramount importance to perform a comprehensive evaluation, able to combine the maximization of pathogen inactivation with the minimization of environmental externalities as ecotoxicity and byproducts and process costs.